see. Okay, so we'll see how we go, because I don't, we should be on the speakers, you see, because, for example, if one of the people, Sullivan, would you mind turning your mic on and saying something?
Hello?
Oh, so that doesn't sound all connected either, does it? So something isn't connecting up. And I don't know, because here is the speakers. Oh yeah, a link to the speakers. Same.
Now we can't hear you, I think. No, we can't hear you now. Wendy, we can't hear you. You've turned your speaker, your sound off. No. You're muted now.
We can hear each other very well. Whoever's speaking.
Yeah, we can hear each other well, but we can't hear Wendy now.
Yeah, and she wasn't hearing us well apparently either earlier.
Yeah, now she's clicked something and she's muted herself. I don't know if she can even hear that I'm saying this. I don't write in the chat.
Hello?
Oh, yeah, you're back.
Right. Okay, so we're all here now. I'll hear you, and it's really loud, which is fantastic. Okay, great. I'm really, really excited. I need to remove the chat somehow, but I don't quite know how that works. But
just hit the next at the top right.
But then I'm going to close the whole thing.
Yeah, no, so the down arrow.
Where? That one? That one.
Okay.
Yay! Right. Okay. Really, really excited to introduce to you Professor Rana Dajani, who is a really exciting person who I've been aware of for a few years. She's a professor of molecular biology. She also is the founder of the We Love Reading program. She's based at different universities across the world, but your main base is in Jordan, isn't it, Hashimite?
Yes.
So welcome here and looking forward to your talk.
Thank you. Thank you very much for inviting me to share some of my experience and my work. And I'm here also to learn. So please write down your questions, your comments, most importantly, your critiques, because that's how we can grow together. So that later on, when we have a session for Q&A, I'd be delighted to interact with you. And I want to thank Wendy for inviting me. And I want to thank Helen, who actually brought us, I think, all together. And I met Helen back in 2017 at Harvard. And I'm so excited to be with her again, even if it's virtual. And everybody's welcome to Jordan. Now you know somebody in Jordan. So I'm going to start by sharing my screen. Let's see, this will work. Great. And now do my PowerPoint. All right, can you see it? Great. So I'm going to talk about different aspects of my research, and I do many things. And I like to start by introducing myself by telling a story. And I believe storytelling is very powerful. I'm a biologist, and therefore evolution is a central dogma and theme of everything I do. And I envision somewhere in the past, as humans were evolving, there were two groups of people. There was a group of people who sat around the fire and told stories. And there was another group of people who frowned on those who were telling the stories and told them, you're wasting your time. But I think you can imagine with me which one of those two groups actually survived. It was the storytellers, because we learn through storytelling, and it's such a powerful tool. So in that spirit, the spirit of evolution, I'm going to share a number of stories. I like to introduce myself by saying that I wear 5 hats, because that represent different roles that I've done in my life. And because I don't wear a hat, I wear a scarf. I talk about my 5 scarves. And the first one is I'm a mother. I have actually 4 children. And that's the most role I'm very proud of, because nobody can replace me as a mother. And my other roles, I'm replaceable. And I like to talk about it first and foremost, because that's the best I can do for humanity, is to bring up the future generation. The second scarf that I wear is I'm an educator. I was actually a school teacher for 10 years before I went and did my PhD and became a professor. And I think children, if they're not spending time at home, they're spending it at school. And so the role of the teacher is very important in bringing up the next generation. So my hat off to all teachers. Thirdly, I'm a scientist. I'm a molecular biologist. And my talk today will talk about my science, which, as you saw from the title, I study trauma, not just the impact on behavior, but on physiology and ultimately on DNA in the form of epigenetics. I also study ethnic populations. So we're the world experts on the Circassian population and the Chechen population who come from southern Russia. And my work in understanding different diseases in these communities led me to work on stem cells. And working on stem cells led me to ask myself an ethical question: is it okay for me to work with stem cells as a Muslim? And when I didn't find enough information out there, I actually spearheaded an effort committee to create the first law that governs stem cell regulation and therapy in Jordan, and actually in the region. My fourth scarf is I'm a social entrepreneur. And I started We Love Reading, which I will also touch upon, because finally I have been able to marry my science with my social work, entrepreneurship as well. My fifth scarf, I'm not going to tell you what it is, and I invite you to read my book, which I wrote when I was at Harvard, where I met Helen. And Nature actually did a piece on it in 2018. So, and I can share with you the PDF of the book because it's quite expensive. It's been considered like a textbook. So I'll send to Wendy the PDF and you can of a review copy, and you can read it, enjoy it, and tell me what you think, because I'm on my second book now. And there's an Arabic translation as well. So, my research, I believe a scientist is seeing what everybody sees, but thinking what no one has thought. Not just a scientist. This is part of human nature. And as a scientist working, as I said, with Circassians and Chechens as ethnic populations in Jordan, the Syrian crisis happened in 2011. And Jordan has always been a haven for refugees from across the region. First, the Palestinians, who, when Zionist forces took over Palestine, they fled. My father is from Palestine, from Jerusalem, so I'm a daughter of a refugee. And then the Iraqi war and all the refugees from Iraq, and now the Syrian war and the Yemeni war and the Somalis and Sudanese. So Jordan is a haven. So being a scientist and seeing in 2011 the Syrian crisis happening, and by the way, my mother's Syrian as well, from Aleppo. So again, a daughter of two communities, but I hold a Jordanian citizenship, and I'm not alone. So many people in the region come from multiple backgrounds. It's actually colonization that actually created the boundaries and borders that didn't exist 70 years ago. Jordan, Syria, Palestine, Iraq was all one big Levant, the Levant. So these kind of passports and citizenships are a little bit artificial, and I'm an example of that. But my curiosity was piqued because most of the refugees, not just from the Syrian crisis, but actually worldwide, are children under 18. And to give you some statistics, there's actually 68.5 million people forcibly displaced. One person displaced every 2 seconds. Just imagine that. And now we have the Ukraine crisis unfolding as well. And then if we look at the how many in Jordan, because I'm living in Jordan, we see there's, and these numbers need to be updated. We're almost reaching 1 million now in Jordan of refugees from Syria. Now the impact on mental health in terms of the children, we know already that 45% have PTSD, 20% have been clinically diagnosed for depression, and many, many other things. I'm not going to spend much time on this data, and I'm sure you can find them anywhere. It's a big challenge. And in the past, people would focus on food, basic health, and shelter. And now we've realized that we need more than that. We need access to health, education, freedom to work, employment support, and not to live in camps, actually, but to live within the host communities. And Jordan is a prime example of that. 80% of the refugees actually live in host communities. And this is also a shout out. Everybody who wants to study refugees say, I want to go to the Zaatari camp. It's become this kind of the thing. when most of the refugees are actually outside the camp and need help, because those inside the camp are served very well by all the international NGOs pouring in. They suffer from other problems. I'm not saying the camp life is great, but each one has its own challenges, and it's good to be aware of that. So what we wanted to do is understand which interventions effectively really promote health and abate violence, and how can we design good research to get robust evidence to evaluate these programs. I mean, there's international NGOs, again, are pouring in, money's being pouring in into Jordan to serve these refugees. And we've got all these interventions, from Brazilian dance to yoga to, you know, you name it, somebody's come up with an idea and wants to implement it, all with good intention. But the refugees have the right to know which one works and which doesn't, and to have some ownership and maintain dignity. So in that spirit, we wanted to evaluate this program, see what works for whom, right? So we wanted to look at the quality and depth of the evidence regarding humanitarian public health programming globally. And we used Jordan and Syrian refugees as a prime example. So we partnered with Mercy Corps, who had developed an intervention, which they called No Lost Generation, which they had scaled to almost 500,000 war-affected youth in the region, right? Syria, Turkey, Iraq, Jordan, Palestine. So this is a psychosocial intervention of 8 weeks, and it was deployed to Syrian refugee teenagers and Jordan hosts as a comparable group from vulnerable communities of the Jordanian population. And in these 8 weeks, you either take a carpentry or a hair, barber activity, or computers or sports, different kinds of activities where you worked with the other teenagers as well. So we wanted to evaluate this program, and we did a randomized control trial on it, and we did 3 time points: baseline before the intervention, 8 weeks after the intervention, and then a year after, to see the longitudinal impact of the program. So the first thing we used to assess the impact of the program on mental health was self-reporting. So we had trained Syrian and Jordanian research assistants to gather the data through interviews and surveys. But we also, and then we published this, and we showed actually that although the program claimed to alleviate stress and reduce anxiety and promote resilience, we found that it actually, yes, it did reduce stress and anxiety, and we published this in the Journal of Child Psychology and Psychiatry, but it did not improve resilience. Which, in a way, it's good. If you found everything improving, then you start doubting the research. And but if you have some that are improving and some not, then you can trust the data and the research. But more importantly, this focus on resilience, which the program had claimed, we actually devised a whole new tool, which also was published in Arabic, specific to Arab refugee teenagers, to really measure resilience, and in order to identify if this program works. And the people who actually developed the resilience survey or tool were the Syrian and Jordanian research assistants. So it came from them. When they were doing the self-reporting here, they saw that the kids, after measuring trauma, anxiety, depression, were coming out of these interviews feeling really depressed just by going through the survey and self-reporting. So when we added the resilience tool, they came out of this, happier, because it was asking, how do you cope? What makes you happy? And things like that. And this is, I'm highlighting this because to bring your attention to the importance of the local people to be involved in the design of the research and assessing the tools that are going to be used and making sure that they're culturally relevant. And so actually Science ran a whole special issue on resilience, as you can see here, and our work was highlighted there. And the cool thing is that the reason that this program, the Mercy Corps intervention, didn't work for resilience is because it was designed in the West by Westerners, and it focused in the design that if you're going to build resilience, the concept was built around the individual, because Western societies are more individualistic, while in our societies, Arab societies, we're more about family and community. And so what we found when we measured resilience is that they built their resilience by interacting with their families and community, and the intervention did not have a family component or a community component. It was only focusing on the individual. So now they took that learning and actually changed their programming to add different activities that involve the family and the community so that they could improve resilience as well. So these are lessons learned on how you design research, how you integrate with the people. Now, we also, self-reporting is not always the best tool to really assess the impact of trauma. And we know that your body doesn't lie. And actually, when I went down and talked to the Syrian and Jordanian families, I was saying, I'm half Syrian, so I'm from you, I know you, and I'm I'm a Jordanian, I live in Jordan, so the Jordanians relate. And I told them, you know what, we're gonna evaluate this intervention, and we're not just gonna do self-reporting. We're gonna see what your body says, because you may self-report, but it doesn't really portray what you're doing. And they really got excited. And I said, this is for you to have some agency over these programs to make sure they work, so that you have a voice. And I said, we want to go to science to see how can we measure your resilience or your stress inside your body. And everybody got excited about the science. So this was a science lesson for the parents, for the children. And we told them how cortisol is a biomarker that measures as an indicator of how stressed you are. And that the more stressed you are, it goes up. And if you're less stressed, it goes down. And we told them there's many ways to get, to collect cortisol. We could take a blood, but everybody said, no, we don't want any blood. We said, okay, we could take saliva. And then we tried that with the families and they said, no, that's too messy. And then I told them, but science says cortisol is deposited in your hair. And everybody got excited. And I said, but do you think we could do hair? So they were my consultants. They were designing with me. And they said, yeah, we could do free haircuts and get that sample of hair anytime. So that was cool. And so what we did is we gathered hair strands, 100 strands of hair from the back top of the children's heads. And we were able to measure cortisol because every month your hair grows 1 centimeter. So you have a record of how stressed you are as long as your hair is. The funny thing is that the boys, when they came to the intervention, right when it was time to take especially at baseline, they all had a really nice haircut because they were coming to this intervention, which is, oh my God. So we had to call them back and say, don't cut your hair because we need your hair. And we were able to show that this intervention indeed reduced cortisol as an indicator of stress and anxiety. And we published our work in psychoneuroendocrinology for those who are interested to read more about it, and the complexity of the data, of course. And this is the team that worked on this part of the research. And it's a huge collaboration led by Yale, funded by ELRA, and we had Harvard University and University of Ontario in Canada as well. Now, we did other experiments, of course. We looked at cognitive function and to see what is the impact of war on cognition, and we didn't find any. And the funny thing is that we found that poverty had actually a larger impact than trauma and war on cognitive function. And that's a huge lesson that was published as well, that how much we need to focus on poverty for everyone as well. But we were also interested in looking at DNA, the impact of trauma and stress and the intervention on DNA. And why is that? Because we know that the environment, who we are and our phenotype and how we behave is a combination of our genetics and the environment around us. So we wanted to look at that interplay. So we chose to look at a particular gene, MAOA, and this is, it's an enzyme that breaks down neurotransmitters, including dopamine. And of course, dopamine is linked to behavior. And this is X-linked, meaning it's because the males have one copy, you see the impact of it only in males. So we studied the male teenagers, both Syrian and Jordanians, to see if there's any relationship between MAOA amount, activity, and perceiving stress and resilience. Now, the unique thing about MAOA is that it has many repeat polymorphisms, so many copies. Some individuals have only 2 to 3 repeats, and those are considered to have low activity because they produce a low amount of this enzyme. And some have more, 3, 5, 4 repeats. And for those, they produce more of the enzyme, and therefore they're considered to be high activity variants. And as I said, in the past, there's been reports, research showing that these different variants are associated with altered behavior when combined with early life stress. So those with low activity have environmental sensitivities. So what we did is we found in our cohort, comparing using the Syrian and the Jordanians combined, that over time, with the intervention, over time we found that the children's perceived stress actually reduced for those who had for those who had a low activity of the MAOA variant. And that's very interesting. But then we wanted to add a third component, which is resilience, right? To see, does resilience impact the perception of anxiety and trauma and stress? And does that differ with the type of MAOA variant you have? And what we found is that resilience had an additive effect with MAOA on perceived stress, okay? And as you can see, the blue line is those that have high resilience and low activity of MAOA. So this was interesting, and we published it in PLOS1 for those who want to know more about it. So that was looking at the genetic background of these children and to look at the impact of the resilience and the stress. And this reminds me of the orchid hypothesis. I don't know if you're familiar with it. So this was proposed now almost a decade ago, showing that if someone is an orchid, they're highly sensitive and have to be in the right environment. Otherwise, they'll wither as any orchid. And so when we look at children and we try to understand them, we look at their genetic makeup to understand how they interact with the environment. So if they have the genetic makeup of an orchid, that means they're very sensitive. Meaning if you give them an intervention, they will respond better. But if you also, if they undergo stress or trauma, they respond very bad. Okay, so it's not about a good program or a bad program, or bad life experience or good life experience. It's also how sensitive you are to the environment around you. And then you have on the other side of the spectrum, the dandelions, who are tough, right? So they adapt to any situation, so they can survive, trauma, stress, and they can do good with any intervention that comes. And then tulips are everybody who falls in the middle. So this adds to the picture so that we can understand better how the children are impacted by their environment, whether negatively or positively. And of course, the work done on the MAOA was done with also Yale and University of Florida. And that's the team that worked on that. A movie was made about our work, Terror and Hope. It's a documentary, The Science of Resilience. And I urge you to look it up, and if you want to screen it. And it brings to the fore how we always talk about war without talking about science. So in this movie, we were trying to show how good science can inform interventions and can inform how to do better science. Not to say that the most important thing is to stop the war altogether, but with what's happening, this is one approach of how to reduce the impact of war. Now I'm going to come to the last part, which is epigenetics. So we wanted to go further, right? Not just study the genetic makeup, but to look at the interplay between DNA, your environment and genetics. And that's what we call epigenetics. And that's what finally gives the phenotype, how we behave around us. So cells listen, not literally listen, but they can sense the environment around them, because something in the environment of the cells changes, and this results in changing of receptor three-dimensional structure that relates a message into the cell, and the cell responds either in a fast response by moving or secreting something, or a long-term response by changing DNA expression. I don't know how much your biology background is, but DNA is DNA, doesn't change, but which genes you turn on and off may change. And that's what epigenetics is all about. Your environment can turn on and off genes. So this is your DNA, and it's wound around proteins. And it can have little molecules that are added either to the DNA or to the protein that helps in folding, that actually makes it turn on and off a gene. By folding more, you're turning off a gene because you can't access the DNA. And by unwinding, you're turning it on. And what helps in the winding and unwinding, or access to the turning on and off, are these molecules. And those molecules, binding or not, are a result of the environment outside the cell. So what we found in mice is that, for example, a mouse that grew up in a very stressed environment compared to a mouse that grew up in a less or low stress environment, when they grow up and they have their own pups, the mother that is stressed will not take care of her child. It won't lick it, it won't nurture it. That pup will grow up to also be stressed and not lick its own child, versus the mother that grew up in a not a stressed environment will take care of its pup and lick it and take care of it. That pup will grow up to take care of its own children. All these mice are genetically identical. All right. So it's not the DNA, it's the environment around them that's different. And because it's transferred to the pup, so the pup did not experience trauma, yet it behaved just like its mom. And not that the mom was there doing it and the pup was learning. These are mice. You remove the mom, and we found that the pup kind of portrayed that behavior. And for this particular example, tying it back to cortisol, what happens is that, you know, the cortisol in the mouse experiencing trauma is secreted. And because you have a receptor that binds it, the glucocorticoid receptor, then it reduces the level of cortisol in the blood and everything goes back to normal. Now, for those pups that were not licked, the gene for the glucocorticoid receptor was turned off. So they don't have the receptor. And so the cortisol in their blood remains high. So they're always alert and stressed. And the pups where the glucocorticoid receptor was not turned off because the mother was taking care of it. So when they got stressed, the receptor removed the cortisol from the blood and it went down. And of course, this anxious behavior being transferred or this environment affecting the pup, one would ask, why has it been evolutionary conserved? Why didn't it go away by natural selection? Well, it has its own advantages, because if the mom was experiencing a stressed environment, you would want the pup to also be ready to deal with that kind of environment. And so be alert in case something is going wrong. So it's always going to be selected for to a certain extent. And of course, we know this is epigenetics because we know it's reversible. Meaning, if I take a drug that removes those groups on the DNA, that little molecule, I can make the pup that was anxious to become less anxious, or a pup that was not anxious to become anxious, just by adding that chemical. Of course, we can't do this in humans. Those experiments haven't been done, but we can, project that humans have similar mechanisms in place. So that brings us to the humans. So humans, as they grow from a zygote, growing in utero, in the mother, and then being born and growing up, they have the same DNA. So it's the same genome. What changes is your epigenome, meaning what genes are turned on and off because of the environment around you. And therefore, there's different types of exposure. You could have maternal exposure, meaning the mother eating and drinking and how that affects her fetus, the fetal environment in utero, and then as you grow up as an adult, what's happening. And so if we were to look at the disease risk over a lifespan at the bottom from early to mid to late life, we see the different factors, as you can see here, that impact the disease risk. So whether it's your race and ethnicity and the environment you're in, or it's the different practices, you're educated, how much money you have, you're on drugs, et cetera, where you live, or which are the social determinants of health, or the epigenetic modifications that occur because of those social determinants and how that affects it. So, oops. Yeah. So, and the importance or tying. So one thing is that when we talk about stress, we're always talking about negative impact, about trauma, and rarely we talk about positive, which is resilience. And saying that I want to see the impact of war and trauma on children or adults and looking for a positive response, not a negative response, is new. And when I started talking about this 10 years ago, everybody looked at me kind of like, Rana, what are you talking about? You're encouraging people to go, you're encouraging these kinds of experiences because you're promoting a positive response. I said, but this is a reality and we've evolved and survived. I'm sure there's not just a negative response. There should be a positive response on how to cope. Nobody listened. And again, whenever you come up with a new idea, people are going to say no. And then last year, Michael Pelusi and others wrote a review on the role of epigenetics and psychological resilience, which is exactly what I was talking about. So if you have a good idea, write about it, because, and then later somebody would claim it, because this was a review and there was very few research tying epigenetics with resilience and war. And they were just trying to summarize and proposing this new, let's say, kind of hypothesis. So read it and trust your gut feeling. So as you can see, with time, we have a biological clock, which is measured by this epigenome. Adding these, turning off genes, turning on genes. That kind of is a measure of aging, right? And so with certain behaviors, we accelerate aging through this epigenetic impact. So we increase risk to cancer, other syndromes and diseases or infections. And this differs between males and females. And so we can actually de-accelerate the aging by changing our lifestyle, our nutrition, and so on, or interventions. So for a clock for pediatric, a pediatric clock was developed by Michael Kober at British Columbia, Vancouver University. And this, it's called a pediatric buccal epithelial clock because you could just take cheek swabs and measure that clock looking at the epigenome. And this clock is impacted by intrinsic factors, right? Who you are, you're born with your genetic makeup, and also by physical and social environment, and by interventions. And so we wanted, and this impacts you all throughout your life. So we wanted to use that clock to measure the impact of an intervention, because what's lacking, and this is a very new field, what's lacking is looking at the impact of interventions and looking at longitudinal impact or longitudinal progression of this clock in different communities, not just Western-based communities. So this leads me to We Love Reading. So We Love Reading is a program I started in 2006, and it came from a feeling of responsibility to employ the skills and my knowledge to solve a problem in my own community, beyond my classroom, beyond the lab. And I realized children don't read for fun. And it wasn't just Jordan, the Arab world, it's actually all over the world, even in those developed countries. And so I found out that to foster a love of reading, one has to have a role model. One has to read aloud to the children as early as when the mother's pregnant. Because as the neurons make connections, there's an association, a feeling of love and security that's associated with reading that stays with you when you grow older. And so from that premise, we started training women, men, and youth how to read aloud as an art in your native language on a volunteer basis in your local community from the local culture. And that program, we called it We Love Reading. And you can see the picture that Menif says without words what it's all about. And the impact was on these adult volunteers, on the children, and on the wider community. And we were not just impacted reading and education by, these kids got really excited about reading and wanted to go to school. And, if you love to read, you love to learn, and you become a lifelong learner, especially with COVID, this became a fantastic placeholder. But also what we found is that it reduces mental health and boosts resilience because you're sitting with an adult, you're reading aloud a story. And this human-human interaction is so important, to lose yourself in a book and forget the world around you. We also discovered that it's about leadership in our volunteers, who are mainly women. So they now had a mindset of I can, and literally found their voices, literally by reading aloud and figuratively by becoming readers. So now we have a global network of people at the street level. And especially with COVID, this became so important for all those reasons. And we were able to address multiple SDGs, and we spread to 63 countries around the world. And we're still spreading because it's a social movement, and it's based on such a simple concept, while serving universal values. So because I'm a scientist, I did not rest that, okay, I got a great program that has spread. I wanted to do research on it. I wanted to understand its impact and to develop it better. It's not my field of expertise, so I invited scientists from around the world. So Professor Dima Amso from Brown University actually studied executive functions in these kids before and after the intervention, and showed that all executive functions, working memory, inhibitory control, and emotional regulation improved. We also worked with the University of Chicago and showed that empathy actually doubled. And we're actually working now with NYU Abu Dhabi, looking at mother-child relationship using fancy Google glasses and blood pressure, as well as cortisol, to see the impact on the mother-child relationship and how that impacts physiology. New York Times wrote about us. We just won the Schwab Award for social entrepreneurship this year in February. And a documentary was made on one of our We Love Reading ambassadors, Esma, from the Zaatari camp. And this is coming out. And if we have time, I can show you the trailer. And of course, we created children's books. And we started pushing through messages, through reading aloud, to change children's behavior. So we have a whole set about the environment, about gender, about disabilities, and about refugees. And this is a shout out to you. We work with other universities with their students to develop children's books, because we have a whole department for book development. And so this became a nice exercise for students. This is from NYU Abu Dhabi, where they developed a children's book in English and Arabic to serve the wider community while improving their skills, and therefore fulfilling the objective of the university to produce a citizen who can make a difference in the wider community. And of course, now the ultimate, which is the marrying between biology and we love reading, is we're working now to measure this pediatric epigenetic age of the children who are involved with We Love Reading to see, does We Love Reading as an intervention actually decelerate the aging clock for children who have experienced trauma, in this case, Syrian refugees. So we're doing a randomized control trial at this moment where we want to see the impact of the intervention while controlling for socio-emotional development and other genetic, predisposition. So we randomly assigned a group to We Love Reading and those not, and then those who aren't taking the program, we'll give it to them later. And we're doing 3 time points because I told you the important thing is to look at interventions now and longitudinal impact of interventions on epigenetics. And lastly, I will end now, the question that that's burning now is, after we understand, and we already know in animals and we're proving in humans that the environment has an epigenetic signature on a human being within their lifetime, the question now, is that transferred across generations? We know it happens in animals and mammals, but we don't know if it happens in humans. We know that 1% of genes escape erasing the epigenetic signature through a process called imprinting, but we don't know about humans. And that's the $1 million question. I don't think it's a $1 billion question. So what we're doing now, and we just got a grant from NSF, we wanted to look at 3 generations of females, their grandmother, the daughter, and the granddaughter. Or you could look at it, the mother, the fetus, and the reproductive cells, the eggs. If this is a female fetus, her eggs are developing at the same time. And so we wanted to look at the epigenetic inheritance across 3 generations. And because I'm Syrian, I was able to identify cohorts of families where the grandmother was exposed to trauma in 1980. There was a huge massacre in one of the cities in Syria, or the mother was exposed in red during the 2011 war, or non-exposed Syrian families who were never exposed to war. And then to compare those 3 cohorts, taking 3 generations of females and comparing them to each other. So we finished collecting the data, we finished doing the epigenetics, and now we're actually doing the analysis. And my daughter calls this the immortal kiss of our grandparents. And this is the team working on the epigenetics of transgenerational trauma. Again, we have Yale, Catherine Panterbrick, and Connie Mulligan, and we have the NSF grant. So everyone is unique. Nobody has a DNA similar to anyone else. Each one of you is unique. And even if you are identical twins, your epigenetic or your epigenome is different because you're slightly experience different environments. So you have something to give to the community. As students now who you're going to go out in the world, remember that. And if somebody thinks or tells you you're just a drop in the water, tell them, but what is the ocean except millions of drops? So yes, you will make an impact. And my motto is the butterfly effect. So be the butterfly. When a butterfly flutters its wings, it moves the wind a centimeter. There's a hurricane in time and space beyond what we can consider. Thank you very much.
Thank you so much. Thank you, Rana. Can you hear me at the moment?
Yes, I can hear you.
Fantastic. Are there any questions in the audience? We have 10 minutes for questions, if anybody has any question. Let me just see if you can probably see us as well.
Yeah, it's not a question. It's just wondering whether we can have this to see the slides later, so we can look at all of the slides.
Definitely. Yeah, I'll send them to Wendy. That's pretty huge. I'll send a WeTransfer and then you can share them.
Good.
Thank you. Thanks. So I have a question. This is really exciting and thanks ever so much. Really, really interesting. And I'm sure the students really enjoyed this as well. So we are running a module called Approaches to Interdisciplinarity, and it's really quite exciting to see how your background is molecular biology, and then you've gone into We Love Reading, and so I'm interested in your sort of viewpoint on interdisciplinarity and how you work, for example, with people from different disciplinary backgrounds, but also how you are interdisciplinary within your own work.
That's a great question. I think going forward, in science and in solving or achieving the SDGs, the only way forward is interdisciplinarity. Now, how do you do it, right? You have to be strong in your own discipline to start with, because that's where you get the confidence and the courage. But then your curiosity gets the better of you. And you start saying, okay, oh my God, that's really interesting. What does that mean? Many times they tell us, no, stay within your silo, stay within your field. Don't go and ask another question you don't know or you don't understand. So I don't listen to those people. And I let my curiosity guide me and I go ask. Now, the cool thing is, because it's not my field, I have a license to be naive. I have a license to ask a silly question. And that opens up a whole new world, not just for me, to keep that curiosity moving, but also for the other field. Because usually when you're in a field, you're so much into it that you become so narrow-minded and so limited, because people, as you went through the field, told, oh, that's impossible. You can't do that. The theory says this. But by the time you're a professor, all that has changed, but you haven't realized you're still stuck in your old thing. So when somebody naively comes in and starts challenging you, that is so helpful and so healthy. And it makes you step back and say, yeah, why did we say that theory, or why, or who said so? And I'll give an example from my field. So I grew up, I went to the university for my undergrad and graduate in the late 80s. And at that time, there were an adult cell was an adult cell. It could never go backwards and become a stem cell. And if anybody even suggested it, people would look at them like you're crazy or you're really ignorant, right? And then the scientist from Japan, Yamanaka, said, who said so? Why can't I reverse it? And he had the courage and the guts to go against the dogma, and he found a way, and he won a Nobel Prize in 2012. And so, and many are like him. So it's about trusting your gut feeling, doing your homework and doing the research, but keeping that curiosity lit inside you, and the courage to ask and to challenge those around you. And if you're in the other field, if somebody asks you a challenging question, to take it to heart and think, oh, wow, I never thought of that. Let's explore this together, and not to feel threatened at all. But this is the way forward, because innovation and creativity happens at the boundaries. And that's what I found. It's that boundary between we love reading and my science that created that creativity in what we're doing. I went into the mental health field totally naive, but I felt I got a whole nother PhD, but at my leisure, because I wanted to. And I was asking all these questions and writing all these papers, and it was a whole new world, but it brought out new ways of doing things, new partnerships, and it advanced the field because coming in naive. And lastly, it also enriched me as a person. Sometimes you get so frustrated from your work, things are not working. And so what I would do is I'd go and work on We Love Reading. And then when I get frustrated with We Love Reading, I'd go back to work on my science. So it's a great break going back and forth between both, and stimulated moving forward all the time and being positive.
Great, thank you. Thanks. I can see there's another question. I'll just stop recording as well.